Okay, so how's it coming with the DNA origami? Some of you look like you're getting it. Some of you are struggling. That's okay. Take your time. Take your time. If you can't figure it out, it's just a fun activity to
Just 100 points.
challenge your brain.
So if we have the midterm, can we either take the midterm or just do this from memory with like loosely duplicate?
So like this will be the midterm. Like you have to fold the DNA origami.
I want the midterm when I turn over the backside to be make a DNA origami strand. 25 points.
There you go.
Now fold this
for
piece.
2.5 weeks, but they're really only a week and
No, really?
Yes.
Wow, we're really getting there then. That's because we cut out a week so that convocation, or is it convocation or graduation doesn't fall on Mother's Day?
Graduation.
Yeah, graduation. So it is, so graduation doesn't fall on Mother's Day. That's why.
Mother's Day last year.
It's usually on Mother's Day.
That's why they did.
Yeah, but it's not going to be. So we're gonna have this weird week between midterms and spring break.
Yeah.
So I hope I actually have people show up to class.
They have to.
Yeah.
Okay.
We're all in Sharpies on doing.
Okay, so we're gonna continue talking about DNA and RNA today. Talk a little bit more about some of these early experiments that have helped us understand the structure and function of DNA.
Now I turn the paper over. Look we go.
I think I missed the section on how to properly do origami in this class.
I thought that was just a
life
life
skill. About basic incision repair? Excision repair? I do not.
I think I'm folding it wrong.
So,
I get it.
hey,
I don't
very
think I did
nice.
it.
This was not
Yeah.
worth it.
Oh, that makes me sad.
Oh, I'm sorry, it was worth it. No, I'm kidding. No, this is kind of good.
All right. You want to walk over and show it to the camera really quick?
Yeah.
So I just found this online. You can Google it, DNA origami, and you can print it out for yourself if you want to or want to do it with kids. We had finished talking about Rosalind Franklin and her ability to get this X-ray of DNA, and we discussed why it may have been wet and why they used it wet, simply because of the possibility of hydrogen bonding and greater alignment of those fibers that they were using. The book does say that they stretched them. They stretched them to try and make them more linear to essentially be an enhancement method to get the image the way they wanted it. I'm not certain how they actually stretched it, whether it was a physical process. I'm assuming it might have been. But I find that very interesting that they said they stretched the fibers. So between the base pairs, every rotation is approximately 36 degrees based upon the, or in respect to the next base pairing. I think it's very fascinating that we also now know that there are multiple forms of DNA, both an alpha form and a beta form, and we'll talk about that and what the differences are between those. But the double helix is our secondary structure of DNA, and today we're going to talk about the tertiary structure and what that looks like and why that happens. So the base pairs are approximately 340 picometers apart, which is 0.34 nanometers. I do want you to know that value, so 0.34 nanometers or 330 picometers between each base pairing. The double helix has both major and minor grooves. The minor groove is the sugar backbone where you have all of the sugars and the phosphates that line up all the way around. Watson and Crick actually didn't know which way DNA spiraled, whether it was a right-hand helix or a left-hand helix. And they guessed it was a right-hand helix, and they actually guessed correct. So once they had, I'm assuming once they had the X-ray crystal structure of this, then that was confirmed, proven. The major groove has more accessibility to the base pairs, and they are more visible there outside or within the backbone substructure. Once they figured out that it was a two-strand model, you heard James Watson on the TED Talk. What did he say? What did he say when they knew they were right? What did he say about copying? That they thought that it was semi-conservative form of replication, but they really knew they had discovered the copying mechanism simply by understanding there were two strands connected. That one strand was going to be used to essentially copy the next. Okay, so you're going to end up having a parent strand and a daughter strand and newly synthesized DNA as a result of this type of process. Okay. Now, they didn't know anything about the exact mechanism of how it happened. We now know that there are proteins and enzymes involved in this. So today I'm going to actually show you a video of a 3D model of DNA replication in real time, as if you were peering into the cell watching it. And it is super cool. It is really awesome. Okay? So there were 3 different possible modes of replication, and they had to determine essentially which one was correct. There was a conservative model, a semi-conservative model, and a dispersive model. And the dispersive model is very interesting. The dispersive model was essentially a mixture of parental DNA and daughter DNA, kind of all mixed together. So Stahl and Mieselson in 1958 decided to do this really very clever density experiment, and I was floored, like just thinking about them designing this experiment and figuring this out, okay? Because they did it by density, and I don't know, I would have not ever thought of doing it by density, but it's a radio labeled experiment with nitrogen-15, which they called their heavy mixture, and nitrogen-14, which is the normal isotope, the most abundant, and that is the light mixture, okay? So nitrogen-15 has one more neutron, so it is a heavier form, and therefore if nitrogen-15 was incorporated into the DNA, it would be heavier as a result. So its density would be greater. So if they had a semi-conservative model that actually occurred, the hybrid model would have a density that's literally in between of the light and the heavy, because it would have one of each. Okay? How cool of an experiment is that? Okay? So they did this DNA density labeling experiment with, again, nitrogen-15 isotopes and DNA from E. coli, and then they measured the density changes after replication. And then they even measured it after 2 generations, just to confirm what that was, or what that looked like. So there were cells grown, again, in the nitrogen-15 or heavy medium, and there were cells grown in the nitrogen-14 medium. And the result, again, was expected if the newly replicated DNA was the hybrid, then they'd have one half the parental and one half of the new DNA to have the density be in the middle. So that's exactly what they discovered and they found, and this literally proved that semi-conservative replication was the mode for DNA replication. So it was a really important discovery that paved the way for other research moving forward. Heaven, did you figure it out?
No, I think we all know why I'm not an art major.
That's okay, that's
Her
all right.
paper's kind of
You
ruined.
can get Caleb to give you a tutorial after this is all done.
To be fair, when I did this in high school, I was also bad at it, and
Oh,
someone
okay.
had to help me.
All
I
right.
tried to help her, but her paper's just too far gone.
Okay, all right.
Get an iron.
Okay. Here is the DNA replication YouTube video in real time. So we're gonna watch this. If you have your book, again, like I was telling you, turn over to page 686. And 686 will show you the enzymes that are involved in the replication process. So you have DNA helicase, primase, you have the lagging strand DNA polymerase and the leading strand DNA polymerase. And you guys get to watch this. And the first time I saw this, I think I was, I don't know, back in grad school maybe when I saw this, but it's really, really pretty awesome.
My essay written, and I've been working on it for about a week. So now I'm going to show you how I use Grammarly to edit.
No
Okay?
thanks.
During DNA replication, both strands of the double helix act as templates for the formation of new DNA molecules. Copying occurs at a localized region called the replication fork, which is a Y-shaped structure where new DNA strands are synthesized by a multi-enzyme complex. Here, the DNA to be copied enters the complex from the left. One new strand is leaving at the top of frame, and the other new strand is leaving at the bottom. The first step in DNA replication is the separation of the two strands by an enzyme called helicase. This spins the incoming DNA to unravel it at 10,000 RPM in the case of bacterial systems.
That's fast.
The separated strands are called 3' and 5', distinguished by the direction in which the component nucleotides join up. The 3' DNA strand, also known as the leading strand, is diverted to a DNA polymerase and is used as a continuous template for the synthesis of the first daughter DNA helix. The other half of the DNA double helix, known as the lagging strand, has the opposite 3' to 5' orientation and consequently requires a more complicated copying mechanism. As it emerges from the helicase, the lagging strand is organized into sections called Okazaki fragments. These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3' orientation. These sections are then effectively synthesized backwards. When the copying is complete, the finished section is released and the next loop is drawn back for replication. Intricate as this mechanism appears, numerous components have been deliberately left out to avoid complete confusion. The exposed strands of single DNA are covered by protective binding proteins, and in some systems, multiple Okazaki fragments may be present.
Okay. That is actually the, not only the best computer model. This is another good one.
These are tiny molecular machines, and
I will show that one to you later, but that one's also really cool. So
Oh,
the,
is that, as Dr. Z put it, those are cute proteins?
those are cute proteins. Have you seen this one before?
Yeah.
I saw the first one like
You-
multiple times.
You saw
Yeah.
the first one that I just showed
Yeah.
multiple
Yeah.
times.
Yeah, he
I
showed that to
love
us.
kindness.
Okay. Beat me to the punch. Okay. That is what is, nice. That is what is happening in your cell real time, and that is even the speed at which it's happening too. So I think that that's also really cool, okay, that we can have those models for you guys to be able to see that in real time. When I first saw that, it blew me away. Like, we can actually develop these models to look at this, just as if you're peering straight into the cell. I love it. Okay. Even during the discovery for Watson and Crick, they knew there were multiple forms of DNA. The alpha form and the beta form, these are slightly different in structure, and we now know that RNA double helix and RNA DNA hybrids can only use the alpha form. Okay? RNA double helix, RNA DNA hybrids can only utilize the alpha form. I want you to talk to your neighbor and discuss why you think that might be the case. And I will tell you that this is the form that Watson and Crick studied. Okay? This is also the form that was crystallized by Rosalind Franklin. Or not crystallized, but observed by Rosalind Franklin. So why? Why do you think only the alpha form was done?
Guys, why is this the case? That is the question.
It seems to me that if you were using X-ray diffraction, the alpha would be a lot more visible because of the tilt on the very ends.
Yeah, because perfectly.
Okay, I'll repeat it again. Why do you think Rosalind Franklin, and why do you think it was maybe easier to see the beta form for the structure?
You mean the alpha?
Did I say the alpha?
You said
Okay,
beta.
nope, I meant the beta. Sorry.
They also need the beta form?
Why? Why the beta form? Why the beta form? Because DNA can exist in the alpha. Now listen, DNA can exist in the alpha or the beta, okay? But RNA can only exist in the alpha. Why? Why? This was actually in the reading.
I'm still working on mine.
They can only exist in the alpha. Maybe it has something to do with the fact that the sugars that you check the DNA libraries for DNA.
Ability of the
Origami.
They're looking on it from
It doesn't have the ribose, which is what makes it out. What's the difference there? I don't know if it's going anywhere. It's just what I think.
Well, but you can have, but at the same time,
Well,
it's alpha beta DNA. It's alpha beta DNA.
Interest.
So RNA, double-stranded, and RNA DNA hybrids only exist in the alpha form, whereas DNA can exist in the alpha or the beta form. But it was the beta form that they observed.
Well, that's probably why.
it's
I
also
think
not.
the reason that would be easier to see because that's what it's tilted.
I'm not necessarily sure they've actually proven why RNA will only exist in the in the alpha form. I'm not sure, but I think they know, or they have a hypothesis as to why.
No, I want to figure it out.
Talk to me, Devin.
Almost. Almost.
No, what were you saying?
I was RNA alpha, maybe because it's easier to pull apart, because on the top many view, it's like they're outside to the sides, which is what the book says.
Oh, okay.
So that's my thinking.
Okay, so I think that's a good reason. It's a good reason. It's not the reason the book gave, but I think it's a good
Well,
reason.
yeah, it didn't why it pulls apart, but it does say the alpha helix faces like further to the outside.
And if that's the case, why is that the case? Why is that the case? And I think that is the result of why it won't do this. I'll give you a hint. Look at the beta form. Look at the beta form. Where are those phosphates? Phosphates are the ones in yellow. Where are they at? They are the yellow ones. Yeah, where are they at?
It's alpha one they.
Here, look at this one. Here, look at this one. This one, that one's yellow. There's yellow ones up there on the outside too. So, yeah, around the peripheral. And they're really more stuck out on the beta form than they are on the alpha form. The alpha form, it looks like they're kind of cockeyed in a little bit more on the alpha form, okay? So, go back. What medium did Rosalind Franklin use?
A wet, bonding.
A wet medium, and why?
Because the phosphate.
What's gonna do to the phosphates? What's it gonna do? What's on the phosphates?
The charge.
The charge,
It's negative charge, right? Yeah.
okay, and what else?
2 oxygens.
Yeah, so you have the possibility of more hydrogen bonding interactions, okay? Well, on RNA, well, here, what's the difference between DNA and RNA? The sugar. So what's on the sugar?
The OH group.
The OH group. Is that sugar really that exposed on RNA? Okay, so what they think that the hydroxy group actually causes steric hindrance within the secondary structure. And as a result of that steric hindrance of having just a little bit more bulk right there at the sugar, that's why it won't actually adopt the other form. I actually wonder if there's any intermolecular hydrogen bonding that occurs on that hydroxy group that hasn't been observed, maybe. And that's actually maybe what keeps it in that form. I don't know. That's just one random thought that I had. But the book does say that this is hypothetical. I don't think they've proven this. They haven't solved this yet. Okay? All right. This is the beta form of DNA on one end. This is the beta from the side end, which reveals local variations and distortions. There's a spine of water, okay, that can exist as a result of those phosphates being on the outside. Yes, ma'am?
Couldn't it also, instead of being steric hindrance,
was thinking more along the lines of, yeah, I mean, I guess that's a possibility. Like an intermolecular, like I was saying, an intermolecular hydrogen bonding. Yeah, I mean, it's possible. Again, I wonder if there's been any experiments to see whether or not they could observe that. You know, it'd be interesting. So now look at the alpha DNA on the end. What do you notice? Let's go back to the beta. Let's go back to the alpha. Look at that.
Bro.
Yo,
I did it!
she did it! Give her a hand! Woo!
Thank you.
Uh,
It only took me like twice as long as it did everybody else.
Take a look. The base pairing is more to the outside. Okay? The base pairing is more to the outside, and that phosphate is kind of tucked in. So I'm going to go back again to the other one, and now look. The phosphates are cockeyed more to the outside, and the base pairs are more centralized and centrally located, essentially. Okay? So I want you to know the differences between the alpha and the beta DNA just from those general regions that we just discussed.
It helps that it's only an alpha, because that makes it more structurally useful. Because
Explain.
things bind in the major group, and because they're so close to the edge, there's a lot more major group than minor group. So there's
Because-
a lot more binding area for binding activity.
So essentially you're saying because there's more room, it's easier to get in there in the major groove?
Yeah.
That makes sense. Okay, it wasn't until 1981 that Richard Dickerson and his colleagues actually crystallized a piece of DNA. How did they do it? Well, they kind of cheated. Okay, they actually synthesized short oligonucleotides in the lab. Okay, so they synthesized these short nucleotide sequences and then created the optimal conditions to crystallize these, because if they're short, they're not going to be as flexible. Okay, so you take away the inherent flexibility of DNA by shortening the sequence. I don't know how many DNA base pairs they use. I think it's right here. Count them up. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Looks like 12. It's like 12 base pairs long. And that was, oh, it says 11. Maybe not. I thought I counted 12. All right, anyway, it doesn't matter. So they synthesized these short oligonucleotides and got the first X-ray, and this is what provided evidence to suggest the DNA is actually bent, and much of your DNA is actually bent in structure. Go figure, DNA is not perfect, okay? We're not perfect individuals. That model does not suggest that DNA is bent, okay? So there are now computer-based models that reveal this similar evidence. Here's another form of DNA, which is kind of a zigzag form called zDNA. I do not know anything about zDNA, okay? Your book talks a little bit about it, and
91.
it's on page 91. Okay, zigzag DNA. A peculiar left-handed structure. Very fascinating. In zDNA, however, the pyrimidines are always anti to one another, but the purines are always syn. That is what makes it zigzag, right there, okay? That those are anti, or the pyrimidines are anti, and the purines are always syn to one another. Very, very fascinating. Abundant evidence that zDNA exists in living cells, but the exact role in vivo is still an open question. So there it is, more research, something to be studied. it'll be really interesting to find out what its function is, why it occurs. Any questions so far? No? Okay. How are we doing on time? We're doing good. This is DNA that has been exploded out or lysed out of E. coli. What do you notice? What do you notice?
It's curled.
It's curled and there's a lot of it, okay? How does it fit in there?
I would say, yeah, it probably like coils back up somehow. I suppose.
That is supercoiling. Yeah, it's called supercoiling, and that's how it fits into the head of viruses and in bacteria like E. coli. There are two supercoiled structures on the slide. These are the top and the bottom, okay? And the middle one is the relaxed form. These are all, what is the circular form of DNA called? A capsid, is that right? What is it? A plasmid, that's right, a plasmid. I was thinking capsid for some reason. So these are plasmids, and again, the middle one is relaxed. Why, so why supercoiling? Why?
I think that's a lot of information that needs to be fit in a not very big space.
Okay, there's a lot of information that needs to be fit in not very big space. There's another reason.
You can't cut it up and stack it, cause that would ruin it.
Okay, you can't cut it up and stack it. Okay, so I guess that's a good point.
Probably protects it from degradation because it doesn't have as much surface area for anything to really.
You can
break it down.
inactivate most of it by supercoiling.
Okay?
Like, not get the expression of it, because if it's wound around itself, nothing can get in to transcribe it.
Yeah, so it's like a compressed form of data, right? Nature's form of compressed data. They're looking all the time. How can we compress data more? How can we compress data more? How can we make data more compressed and still readable? Okay? So they're always looking at this. They've even used or looked at using DNA as a method of storing information. Yep, which is kind of cool. Okay, I just saw a colorimetric assay done by Whitesides, where they have now taken molecular storage down so low that it's just phenomenal. And I think what you're going to see is storage is just going to keep getting smaller and smaller and smaller. You know? We now have single molecular switches. Now, a single molecule that can switch it on or off an electrical signal. I mean, that is freaking phenomenal, right? I mean, just thinking about molecular circuits and how small that can be. Okay. The other thing that I want you to consider is energy, okay? Energy. Strain. You know when we put molecules together in organic and you try and make the three-membered ring, the cyclopropane?
It won't do it.
It will almost break
Yeah.
the tips of your model when you're trying to put it together, okay? So now think about DNA, right? Think about the strain that might occur in DNA in certain conformations. Nature is all about minimizing energy, all about it, okay? So conformations happen as a result of energy minimums. And that's essentially another big reason why DNA likes to do supercoiling and twist up. If you take a look down here at the base pairs per turn, you look at the unstrained circle here that is 10 bases, or 10 units, whatever, 10 turns long, or linking number, 10, okay? And you go all the way down or you go down to 9, and the base pairs per turn is 11.67, which actually happens to be making it more strained. And then you get this twist like a figure 8, that ends up happening. And then you go down and look that the base pairs per turn go back to 10.5 as a result of that. Therefore, the supercoiling released the strain associated with that particular conformation as a result. Molecules do this too. It's really kind of fascinating. If you look at cyclohexane, you're very familiar with the boat chair form, right, of cyclohexane. Now, you go up to, let's see if I can draw it. This will be challenging for me here. I haven't drawn one in years. Cyclooctane. You might think cyclooctane could also be planar because it's so big, right? It's not. What is it? What's it look like?
Is it kind of more of like a, almost a boat shape?
I'm gonna take these away. Yeah, these ends can flip up or down, okay? So, it's floppy, okay? It's floppy, it doesn't like to exist in a plane. So, this also happens with 9-membered rings and things like this. And based upon stability, we actually know cyclohexane is pretty darn stable in its chair conformation, okay? Doesn't want to be in that planar conformation because all the bonds want to be at 109.5 degrees tetrahedral away from one another, okay? So this is all about minimizing energy. That is really all I had for today. That's it. Okay? So I'm letting you out a little bit early. Riley?
Caleb, do we have quiz on Friday?
Yes, you have a quiz on Friday. I don't have my cards come in. Have you heard anything? No? Okay.
I'll have Sandy tomorrow.
Yeah? Okay. So I have a feeling your quiz will be online again. The